adjacent next to, right? Cells directly next to each other. There's also autocrine signaling. What this does is you have this cell produces a protein that is then released and interacts with the receptor on itself. Autocrine, right? Autonomous, it's on itself. What's an example of this? Okay, we think about myotubes, right? Myotubes are muscle cells in a petri dish. Okay, you can grow them in the lab from stem cells and so on and so forth, right? If you take muscle cells and you put them under electrocution, they can contract, they can be placed under load, right? You can stretch and train muscle cells. When muscle cells are stretched or when they're placed under load or tension, they release a protein called mechanogrowth factor. Okay, where does mechanogrowth factor come from? Mechano is relative to mechanical tension. And so, like oh my God, I'm blanking on the word biomechanics. Okay, like biomechanics, right? Mechanical tension. Okay, so when there's tension, the load placed on the fibers, they can sense that. So essentially, these myotubes release mechanogrowth factor that then interacts with the mechanogrowth factor receptor on itself, and then that tells the cell to grow, mechanogrowth factor. So when cells get bigger, muscle cells get bigger, what qualities about them change?
Like more myofibrils?
More myofibrils. What does more myofibrils mean? More force. Does a stronger fiber, is it more resilient to tension?
Yeah.
Yes. Stronger and bigger fibers produce more force, which means that if this muscle cell was placed under this much force, if it releases a protein that tells itself to grow, it gets bigger. Is that same force going to be as stressful? No, because it's a bigger, more resilient, stronger, and more forceful fiber. Right? So if we think about this from an adaptation standpoint, we can actually apply that concept to training. People lift, they get bigger, they get stronger. They get stronger because they get bigger. They can also just get stronger because of not nervous system, but in general they get bigger and stronger. That makes them better at resistance training. Okay? So this is an example of autocrine. It's signaling to itself, right? And then that causes a physiological response in the cells. We have paracrine. We don't really talk about this too much. So juxtacrine and paracrine we don't really talk a lot this semester about, but this is essentially cells signaling to nearby cells, so they release substances or proteins causing effect there. And then endocrine we already talked about in 346 in physiology. You have a tissue or a gland releasing a hormone or a protein that goes into the bloodstream and signals to receptors on distant tissues. So most of the time in the class we're talking about intracrine, autocrine, and endocrine signaling. Okay, so if you had to know the definitions from this slide, those three would be the more important ones. Now, I know this looks like a kidney bean, but it's a muscle, I promise you. It's just my poor attempt at drawing things. So, let's say this is a muscle, it's contracting, okay? The muscle, when it contracts during exercise, it's perceiving exercise as stress. Okay, why does it perceive it as stress? Because, well, number one, if you're using ATP, that's the currency our cells use to do work and to do things with, to survive. Okay, so that's one indicator of stress. You have less ATP, you have more ADP. The cell senses that and says, oh my gosh, we're running out of energy. Okay, that's perceived as stress. If you have more or less carbs, or using more carbs or less carbs, that is also sensed by the cell. And the cell can say, oh, well, if we need carbohydrates and we don't have enough of them, that's stressful, right? It's a greater stress physiologically versus having those substrates available to do the work. Not even, like, you don't have enough calories, you're on a diet, and you need carbohydrates or glucose to have certain steps in glycolysis and the Krebs cycle occur. Oh my gosh, we don't have carbohydrates. I guess we need to get that energy from proteins. We break down the protein, turn it into a carbohydrate backbone, and now we have something like that to make energy with, right? So the muscle cell senses energy depletion because we're using the energy during exercise, and the drop in energy availability or the use of certain substrates is sensed by proteins in the cells. And that will eventually cause the cells to be better at using energy or producing energy in the future. There's other factors like mechanical stretch or tension, contraction. The muscle stretch, that's stressful. Okay, there's literally proteins called stress-sensing proteins, like enzymes. They literally sense the tension and the stress in the muscle. We know that heavier loads or more volume can cause more muscle damage. Okay? When there's more muscle damage, that can result in the release of growth factors and or cytokines. Cytokines are inflammatory. Most of them are inflammatory. So if you think about it, you've done a really hard workout, are your muscles really sore? Yeah. Do they get really swollen? Is there a lot of inflammation? Yes, because that damage is perceived as a stressor to the muscle, right? Damage is stress, actually, and so how does it recover? It releases cytokines and inflammation to help clear up that damaged tissue and also signal and tell other things in the muscle, oh, that inflammation tells the muscle to get better and recover. It turns on stem cells and tells them to help recover the tissue. Okay? We already mentioned that temperature and acidity change during exercise. It affects enzyme function, enzyme activity, but that's also an indicator of stress as well. We know that hormones are released during exercise. Hormones interact with receptors. Those receptors turn on things in the cell. The availability of blood flow. Remember phosphocreatine, there's no oxygen. Phosphocreatine isn't resynthesized. So if you have blood flow available, you can get the glucose, you can get the oxygen. If blood flow is inadequate, you're in a hypoxic state, right? You're in a nutrient deficient state. The cells don't have the oxygen and the substrate to do work. That causes stress, right? Cells literally die if there's no oxygen. So you get that. Related to systemic factors. The nervous system, right, also communicates, and the muscle can tell, like from the nervous system standpoint of view, there's a stress or contraction happening because when calcium is released into the cytoplasm, that allows muscle contraction to work. The only reason calcium was released is because there was an action potential sent from the nervous system. So the release of calcium is really how the nervous system and muscle can sort of communicate with each other at the biochemical level, right? Because if there's an action potential sent, calcium is released, and it's present, it's there, it's available. If it's not, it's in the SR. So now the muscle knows, oh, nervous system is firing, nervous system is not. The nervous system's firing a lot, the release of that calcium will tell the fiber to become more type 1, to become more oxidative. If you release the calcium, but not a lot, not very persistent, you can still contract, but the fibers won't turn to be very oxidative. They'll still stay more anaerobic, right? So it can regulate those adaptations of fiber type shifting. And that all comes from how the nervous system was firing, right? In addition to, if you're using more energy, okay, those energy substrates tell the fiber to become more oxidative, more mitochondria. More muscle contractions, more calcium release. The nervous system and calcium release tells the fiber to become more oxidative, more mitochondria, right? So notice how all these systems and responses to exercise produce stress on the cell, and that stress is culminated into an adaptation that lets that muscle be better for the next time it does that type of exercise. Clear? Cool. So, there's three major cell signaling pathways we'll talk about this semester. Okay, one is the AMPK pathway, which primarily regulates glucose uptake and mobilization. Another is going to be the mTOR pathway, which the mTOR pathway is the primary pathway involved in protein synthesis. There's the MAPK pathway. They only show two proteins here, ERK and P38, but these are part of the MAPK pathway, but they regulate gene expression and also protein synthesis. Okay. Then also there's calcium-sensitive pathways that turn on when calcium is released in the cell. We've actually already talked about some of them, like calmodulin. We talked about glycogen breakdown, right? What regulates glycogen phosphorylase activity? Like calmodulin, right? It's right here. We don't talk a lot about it this semester, but there are some proteins we will bring up here and there, right? We don't talk about it in depth like the other pathways. Yeah, muscles contract, metabolic rate goes up, stress increases, contractile stress, metabolic stress, oxidative stress, and calcium is released, right? So all those factors happen during contraction, which turn on these signaling proteins, which eventually produce RNA here, that RNA is turned into a protein, and those proteins eventually become the adaptations to the training stimulus. From this lecture, you need to know the slide 100%, no questions asked. You need to know everything on the slide for for sure, 100%. Okay. For this kind of material, I would suggest doing flashcards initially, and then looking at how these terms get brought up in other lectures later on, just because we literally talk about these proteins for the rest of the semester. So if you don't get them for this exam, they're going to come up in exam 2 and in exam 3. They're never going to go away. So make sure you understand these definitions now. I would suggest at least doing flashcards for this, because look at some of these acronyms. AMPK, MAPK. What's the difference? You literally just switch the A and the M around, right? So it can get confusing. How is that different from AMP? Okay, AMP is a substrate. It's a byproduct of ATP breakdown, adenosine monophosphate. AMPK does not have a phosphate on it. That phosphate is not being used for energy. It's not the same thing as AMP, right? It's an enzyme that responds to energy use. Yes, it can be phosphorylated, but when it's phosphorylated, it regulates bioenergetics. It regulates adaptations, but its phosphorylation is not going to be using energy like when you add a phosphate to ATP or AMP or things like that, right? So AMP is different from AMPK. AMP is from ATP breakdown, right? ATP to AMP. AMPK is an enzyme that turns on when you break down ATP, right? So ATP breaks down, AMPK senses that, oh my gosh, we're using energy. Now it becomes highly active, okay? And MAPKs are a different pathway. So knowing the definitions and the abbreviations are important so you don't mess it up on the exam. Now, AMPK is one we've already talked about. Remember, I showed you that figure with the table in lecture 1 where they did 4 sprints on a cycle, and then they looked at ATP breakdown and whatnot. AMPK only turned on when there was a significant decrease in ATP in the cell, and that happened after the 4th and final sprint. Right. So once ATP went down, that turned on this enzyme, which senses cellular energy, right? So when the cellular energy goes down, AMPK turns on. And why does this matter? Because when AMPK turns on, it then turns on and activates genes involved in mitochondrial biogenesis. What is mitochondrial biogenesis?
Making mitochondria.
Making more mitochondria. So when we think about this, if your cell has more mitochondria, what's it better at doing?
Above the respiration.
Being aerobic, but also can clear substrates too, right? It's got more oxidative stuff going on to clear those metabolic substrates. So it's better at being aerobic. When we say it's better at being aerobic, but what's more specifically is it?
ATP production.
ATP production, even more specifically. Oxidative phosphorylation. Producing ATP oxidatively. That's what it's better at, right? So if we're producing more ATP oxidatively, that is maintaining the cellular energy status with oxidative pathways, right? So what does AMPK do? If energy status is going down, it turns on proteins and enzymes that help you make more mitochondria, so that next time you do that exercise bout, you have more mitochondria to produce more ATP. So there's less of a decrease in the energy status of the cell. Do you see how it kind of feeds back into each other? Now, PGC1 alpha and COX4, you have to know those. They, particularly PGC1 alpha is on the exam multiple times. So is COX4, but we talk about PGC1 alpha in other lectures too, so make sure you know that for sure. So PGC1 alpha is a protein that regulates making more mitochondria. COX4 stands for cytochrome oxidase 4. Where have we heard of cytochrome oxidases before?
ETC.
Yes, the electron transport chain. This stands for cytochrome oxidase 4. It's the 4th complex in the electron transport chain. So imagine, if you have more of these oxidases, are the mitochondria going to be able to produce more ATP? Exactly, right? So cumulatively, we look at this and say, oh, well, these are things that make the muscle better at energy production, which makes sense because this enzyme sensed we needed more energy. So it causes adaptations to make them more energetically efficient. Now, classically, if you look at cell culture and animal models, when you overactivate this protein here, it inhibits mTOR activity. It inhibits the mTOR pathway. So we already said mTOR is the protein that primarily regulates protein synthesis and growth. Okay, so if AMPK is really active, it turns off protein synthesis, essentially, or blunts or attenuates protein synthesis. Okay. However, when you look at human studies, just because muscles contracted and AMPK is on, doesn't mean that these resistance trained people don't gain muscle mass. Right? So in the cell culture and animal models, yes, if you apply a chemical or you turn on a gene a lot that causes AMPK to be turned on all the time, mTOR is inhibited. There is a regulation there. It does influence this activity. But when we translate this cell culture, like really basic science information and mechanistic stuff, to human studies, we don't tend to see that same inhibition, right? So this is just kind of to show you that, yes, we know that this does affect this, but as it relates to training and performance, there's actually a lot of other things that ultimately regulate performance and adaptation long term than just this protein does that, right? So they've done studies where they did resistance training, AMPK turned on, but they still had just as much muscle growth as all the other groups. Didn't matter if AMPK was on, because the stimulus that turned on resistance training was an even greater stimulus than whatever the AMPK was going on, right? So kind of, you want to shut me off, whatever, stay on, but you don't really inhibit me, that kind of thing. Yes?
A couple of questions.
Yeah.
So does the degree of AMPK activation determine how inhibited mTOR will be?
Yeah, so essentially, if you look here, so on all these graphs, so for the rest of the semester, when you see an arrow pointing to something, that means it activates it in general. They almost always show that. If you see a T-bar, that means a blocking inhibition. So AMPK activates this protein, TSC. TSC blocks mTOR. So if AMPK is being activated, it's turning on this protein, which is turning off mTOR. If AMPK is not activated, there's nothing inhibiting mTOR, so mTOR is inducing protein synthesis. What we'll see is that, just like we talked about glycogen synthase and glycogen phosphorylase, right? One is synthesizing glycogen and one is breaking it down. You never see phosphorylase activity really high and synthase activity really high at the same time. Why? Because they're actually doing two opposite functions. In your muscles, the cell signaling pathways are kind of similar to that in a way that you have pathways that are going to be breaking down substrates, or there are enzymes that are turned on to break down and provide energy. mTOR makes things, it makes proteins. It uses ATP to make proteins, okay? For us to build a muscle, to build a structure in our cells, we need to use ATP to do work to build that thing, that protein. If we need the ATP to contract during exercise, we shouldn't be using that ATP to try to make proteins and enzymes, right? So this is why during exercise, you don't see protein synthesis elevated. Actually, it's just either no change or whatever's at rest, okay? After you stop contracting, then protein synthesis is elevated, okay? Because now we can use the energy to build things and recover. But before, during exercise, we need the energy to actually exercise, okay? So you're not gonna see the breakdown and synthesis on at the same time because they compete for energy sources, okay? So yeah, notice how AMPK is really active. It does inhibit mTOR, but in humans, we kind of end up seeing is like, well, AMPK turns on, at least with resistance exercise. AMPK might turn on, it might regulate glucose use and how glucose gets used during exercise. Remember, AMPK is also something that can tell those GLUT4s to mobilize. Right? Insulin tells the GLUT4s to turn on, but didn't I say contraction turns on GLUT4s? Some data would suggest AMPK is that protein that tells those GLUT4s to turn on from contraction. Right? So you might have contraction turning on AMPK, but it's not like overbearing, it's not like overexpressed, it's not on because it's chemically turned on, it's just on because of contraction. And then you also have the anabolic stimulus of heavy loads. So overall, the stimulus is anabolic, tells the muscle to grow, right? With aerobic exercise, is that heavy load?
No.
No. So with aerobic exercise, you use a lot of energy, but it's not heavy load. So do you see a lot of endurance athletes with lots of muscle? No, because they're not doing the type of exercise that builds the muscle, right? Because the exercise is not heavy load. So mTOR is not really on, but AMPK is really on. So yeah, we become more oxidative, but the muscles don't get very big, and that's actually okay. Yes?
Let's say you had like full activation of AMPK. Let's say you did like you were doing endurance exercise, right? How long would that inhibition of mTOR last?
In humans, it's not really like an inhibition. It turns on. And so what they've shown is that when you do more volume of exercise, AMPK turns on even more. But they've also shown that when you do more volume of resistance exercise, so does the mTOR pathway. So they kind of like balance each other out. At the end of the day, you don't really see that the AMPK itself is really inhibiting the mTOR activity, because mTOR is going to be on because the loads are heavy and there's enough volume that will turn on. They will just be on at the same time. The problem is, like, when we study what AMPK does in cell culture and animal models, they actually use a compound that turns AMPK on 24/7. Or they alter the gene that makes AMPK 10 times more active than what you would see in normal physiology. So we know that, yes, that's what AMPK does, and we know that's what effect it has on mTOR. But in humans, you're not going to keep AMPK on 24/7. You're not going to change the gene to make it 5 times more active compared to every other enzyme. So even though mechanistically we know it works, when we translate it to like translational studies in humans and actual training studies, we don't see that same blunting. And there's been quite a lot of studies showing that if you're untrained or if you're recreationally trained, you can do aerobic exercise and resistance exercise and there's really no inhibition in muscle size or growth, okay? However, if you're very, very well trained, if you're elite, well, now the muscles is very specifically adapted to resistance training. Now when you do aerobic exercise, that's a very different stimulus. It's very shocking, it's very stressful. So we do see with very, very trained people, the more volume you do, the more aerobic stuff you do see a loss in size and strength, right? And even if, yeah, we're talking about muscle size here, for instance, there is something to be said that when you do more volume, it does impair power, right? So even if strength is okay and size is okay, something about aerobic exercise impairs power and velocity adaptations to resistance training, right? So we've got to think like, okay, what's our goal to get stronger and bigger? You can still do cardio, no big deal. Just don't do like a lot of cardio, right? Don't train like an endurance athlete kind of cardio. But still, cardio is good for your health. They've actually shown studies that if you did cardio, muscles got even bigger than if you did not. Interestingly enough, right? Because they actually showed when you added the cardio to the training program, that extra blood flow and vasodilation turned on satellite cells and actually added more capillaries, which supplied more nutrients to the muscle fibers, and they hypothesized why the muscles got bigger than when the group that didn't do the cardio.
Were those like trained individuals?
They were recreationally trained people, right? So it just goes to show they like average 2 years, just 2 years of training history on average, right? But if you're untrained, it doesn't matter. You should be doing both, right? The more trained you get, and we'll talk about this in this lecture, the more training you do, the less window of adaptation you see, but that's more specific your adaptations are. The more specific your adaptations are, you start to see more competing or interfering responses from an alternate type of training protocol, right? So for most people, we're not really gonna see the interference, but for elite athletes, Olympians, right, highly trained, 15 years of training history, that kind of thing, yeah, then we might see some interference there. But also they're already at a really high level, so, you know. Anyway, so that's the AMPK pathway. Now, the MAPK pathway is a separate pathway, okay? There's 3 proteins in that pathway. I mean, there's a lot of proteins, right? But there's 3 main proteins, okay? ERK, JNK, and P38, okay? Now, ERK primarily regulates cell size and muscle growth in response to exercise, resistance exercise, okay? you know, a lot of these proteins and metabolism, they're in all your cells. We just actually end up talking about them as they relate to muscles and exercise, right? But ERK is actually, if we look at breast cancer, prostate cancer, cancers, tumors, what are tumors? They're cells that have uncontrolled division and growth, right? So they are cells that normally should have one cell division, right? But instead they just grow exponentially. When you look inside those tumors, what tells those tumors and those cells to keep dividing is because ERK is unregulated. What does ERK do? ERK tells cells to get bigger. So if you look in tumors, ERK is overactive. ERK is not shut off. Normally ERK turns on and turns off. But in tumors, ERK is on all the time. ERK is unregulated. Right, so this is actually a concept that we're familiar with, like cancer models, right? Why do tumors grow? Because ERK is overactive. What does ERK do? ERK tells those tumors and cells to grow. Okay. Now with exercise, normally in muscle, ERK turns on from contraction and then it turns off. So we're not really concerned about like tumor growth in skeletal muscle because really our muscles want to stay small. We're more aerobically efficient, we're more efficient physiologically if muscles are smaller, not spending all this energy on big cell sizes, right? So normally we don't see ERK and these anabolic pathways on all the time because that's not really the way our metabolism also works. But ERK does turn on from contraction and it does tell muscles to get bigger. Now junk is mechanosensitive. Remember I said mechanogrowth factor, tension on the muscle cells? Something to be mechanosensitive means that this protein can tell how much load is being placed on it. So to be mechanosensitive means that, right, this is load. So the heavier the load, this is P junk, phosphorylated junk. The heavier the load, the higher the phosphorylation of junk, the more it turns on. Okay, it's very sensitive to that. All the MAPKs are mechanosensitive, but junk is the most mechanosensitive. What do we mean by that? Because if you look at phosphorylated P38 or phosphorylated ERK, when you look at load and phosphorylation, as load increases, phosphorylation only increases maybe that. Notice, you increase the load quite substantially, but it didn't increase really that much. You increased even more, well then it turned up, but it's only about half of what you would expect with junk, right? Junk, any increase was literally linearly related to a proportionate increase on junk activity. So this is more sensitive to changes in load versus the other ones. But these are still responding to load as well. They're just not as tightly tuned to the tension placed on it, right? So that's what mechanosensitive means, is to be sensitive to the amount of load or mechanical tension placed on the muscle. Clear? So an example, why do we care about this? This point is not on your exam, but I'm explaining how this all relates. Junk turns on when there's heavier loads. If we think about aerobic exercise and resistance exercise, which one of those has heavier loads based on the muscle? Resistance exercise. When you do resistance exercise, do muscles grow? Yes. Muscles grow larger with resistance exercise than they do aerobic exercise. They published a study 3 years ago that showed that when you took animal models and cell culture, when you had mice do resistance training, but you took out all the junk in the cell, or you inhibited junk totally, you did resistance training. When junk was not there or shut off, the muscles didn't grow. Resistance training tells muscles to grow, right? But if junk is turned off and is not there, they don't grow. But guess what? The muscles actually became more type 1, and they stayed smaller, and they gained more capillaries. Those are all what types of adaptations? Aerobic, right? So when you lift weights, junk senses the heavy load and tells the muscles to get bigger. But when the muscle gets bigger, it also makes it less oxidative, okay? Aerobic exercise, in that same study, they had those mice do aerobic exercise. Junk was not on, but the fibers got more aerobic, more capillaries, more type 1s. When they had the mice do aerobic exercise, but they chemically turned on junk, guess what happened? The fibers got bigger, but they didn't get the capillaries or the aerobic adaptations. So what does this say? This is like, you know, a train conductor, this railway and that railway. It's this switch that tells fibers to become more anaerobic, bigger, stronger, resistance exercise adaptations versus, oh, it doesn't turn on because it's not heavy load, aerobic, better at type 1s, right? Better at endurance training type of stuff, right? So this is why, do you ever see a marathon runner look like Arnold Schwarzenegger? No, because literally their training stimulus doesn't turn on junk, right? Because it's not heavy load. But the fact that junk is not turned on lets those muscles become the most aerobic they can possibly be, which is actually good because they're an aerobic athlete. But resistance training is heavy load. So the muscles do want to get bigger, right? Remember too, practically remember in 346, capillaries bring oxygen to the muscle. If it's a smaller muscle, it requires this much distance to get to the mitochondria. If it's a bigger muscle, it requires this much difference to get to the mitochondria. Big muscles are not good, they're not built for peak aerobic metabolism. Why? Because it takes a lot longer, a much bigger distance to get the oxygen to the mitochondria to perform oxidative metabolism. So runners and aerobic athletes, muscles want to stay small because they're better at aerobic, they're more energetically and oxidatively efficient small. And go figure, if junk doesn't turn on, because it's not a heavy load, the muscles stay small. And more capillaries, more type 1. Right? Do you see how these kind of make sense? Right? This is why you don't see, we talked about the more training adaptations you get, then we might see interference, because all that type of adaptation establishes this phenotype, this type of muscle fiber performance. And then if you're doing opposite kinds, now the muscle says, oh my gosh, this is so new, it's so different. I need to go back to that other type of adaptation because my adaptations here are not going to do it. I can't, it's too stressful. I need to become better at this type of exercise, and it'll try to shift. Does that make sense?
Since junk can lead to like the actual growth, does that mean junk will influence mTOR?
More recent studies have shown that junk can actually turn on mTOR. It doesn't turn on mTOR specifically, but it turns on things that mTOR also turns on. So before we used to think of like, oh, there's these three separate pathways. We always think about these pathways as very separate things, but in actuality, they talk to each other. They communicate to each other. And yeah, a few years ago, they showed that junk actually also turns on mTOR substrates, and they both regulate growth in that way. Now, what's interesting is like, yeah, I just told you, now that's not on your exam, but can we talk, this is like an example about how these things regulate adaptations to different types of stimuli, okay? So that's the important point there. Now, junk, during exercise, it will turn on, and then during recovery, right, so many hours of recovery, here's baseline, 1 hour, immediately post-exercise, 1 hour, 2 hour, 3 hours. Junk will turn on, and then over the recovery period will turn off. So in response to exercise, proteins turn on, and then they shut off in recovery, right? Usually. Some proteins turn on later. So you might see mTOR turn on, but then mTOR turns on P70. So then P70 will turn on at 3 hours. And then P70 turns on another thing. The other thing turns on at 6 hours, right? Because they signal to each other. So normally, junk will turn on and then turn off. But what we see is that when junk is on all the time, when it's constantly stimulated, it actually tells cells to die. It undergoes apoptosis. So states where there's lots of inflammation, cancer cachexia, right? Inflammation, catabolic state, lots of muscle loss, lots of atrophy. Why? Because junk is on from this inflammatory state, and that tells the cells to atrophy and die. Okay, so here's the same protein that can tell muscles to get bigger, really anabolic, but if it's always on all the time, now it shifts its function to become catabolic. But it depends on the signals it's receiving, right? It's regulating muscle size. If they're anabolic signals, muscles get bigger. If they're catabolic signals all the time, it tells the muscles to atrophy and get smaller. die. Make sense? P38 is also in the MAPK pathway. It regulates satellite cell function. Right? Satellite cells are essentially your muscle stem cells. We'll talk about them more specifically in Unit 2. Okay? P38 also regulates inflammation. So when there's a lot of inflammation, P38 turns on and makes even more inflammation. So go figure. Okay? And P38 also regulates oxidative capacity. So P38 in combination with AMPK will tell fibers to become more aerobic. Lastly, mTOR. The mTOR pathway is also mechanosensitive. So mTOR is mechanosensitive. It's also amino acid sensitive. So when you consume a meal, those proteins and amino acids in that meal, as they get digested and pass through your bloodstream, the cells, the receptors on the cells can sense those amino acids. And if there's amino acids there, mTOR will then turn on and then increase protein synthesis. This is why if you have a meal, cells maintain their integrity, right? If you are on a diet or you have a protein deficient diet, what happens to your muscle? It goes down. Why? There's no proteins to rebuild the muscle. Why? mTOR doesn't get turned on. There's no proteins. Synthesis goes down, you lose muscle size. Now, how does mTOR work? mTOR turns on P70S6K. It's a protein that activates a ribosome. When ribosomes get put together, they help increase protein synthesis. Also, the activation of P70 is correlated with muscle growth from training. So if P70 puts together a ribosome that increases protein synthesis, then it makes sense when you turn on P70 during training that has a relationship with muscle growth, because it literally puts together a ribosome, protein synthesis goes up. What is muscle growth? It's all the proteins that have been accumulated from synthesis. Much bigger, right? Makes sense. mTOR also works because it phosphorylates this binding protein, 4EBP1, and I'll show you on the next slide. Normally, this binding protein prevents protein synthesis from being active. But when mTOR phosphorylates it, it removes that binding protein, and now this factor can be part of the ribosome and can increase protein synthesis. Okay? So here's the animation, essentially, right? So we just talked about 4EBP1. Normally, 4EBP1 is when mTOR phosphorylates 4EBP1, okay, it's essentially that phosphorylation there then tells the binding protein to get away from this initiation factor, and then this initiation factor can become part of the ribosome. Now the ribosome and also S6 can become part of the ribosome. So the ribosome is actually, you have hundreds of pieces of the ribosome. Think of the ribosome as like Legos, right? And they're all 5 years old and they're scattered across the room, right? Protein synthesis is off normally because the ribosome is not put together. Once you put those Lego pieces, those ribosome pieces together, once that's there, then you can read a RNA and make a protein out of it. But as long as those pieces are apart, protein synthesis is not happening, okay? So, how does cell signaling working? Well, muscle contraction, growth factors, metabolic stress, all those signals from exercise activate proteins on the inside of the cell. And very much like a relay race, you know the baton, the Olympics, you pass the baton from one person to another. That's how your cells and enzymes in the cells and proteins talk to each other. This turns on this protein, well then this protein phosphorylates this one phosphorylates this one, right? I'm using these as examples, but they talk to each other like a communication. They relay a signal. Chemical messengers, that's what they're called, right? So once they get here, for example, P38, once it's phosphorylated, can then go and interact with a transcription factor in the nucleus. That transcription factor then reads the DNA, produces an RNA. At the same time as you contract, notice that contraction also activates mTOR. Amino acids also activate mTOR. Once mTOR is phosphorylated, it works because it phosphorylates P70, which phosphorylates S6. Once that's turned on, it becomes part of the ribosome. When mTOR phosphorylates 4EBP1, phosphorylation removes that binding protein, this initiation factor can become part of the ribosome. Now you have a complete ribosome. You can read that RNA to produce a protein. That's essentially protein synthesis there. Okay? Do not confuse the terms translation and transcription. Both of those terms are on the exams. Every semester, students confuse the terms with the definition. They get the concept, but they're calling translation transcription. So on an exam, does translation do this? They think it's called transcription, and then they put it and it's wrong, right? So you need to know those definitions of translation and transcription, because on the exam, they get asked in multiple different ways. So don't confuse those processes, right? Transcription is the production of an RNA. Translation is translating that RNA into a protein, right? Translation involves the ribosome, specifically, to make the protein. Transcription involves a transcription factor to make the RNA. Okay? Cool. So, that's essentially protein synthesis. Those steps you gotta know on the exam. Those concepts. So, why do we care about this? Because when these cell signaling proteins turn on, they result in the production of an RNA. That RNA then becomes a protein. And then the more times you do that training stimulus, the more adaptations you get over time. So here's an example. Remember our good friend PGC1 alpha? What does PGC1 alpha do? I don't expect you to memorize it. It's the first day, right? You're talking about it. Makes more mitochondria, right? PGC1 alpha produces more mitochondria. It essentially makes muscles more oxidative. So if we look here, they did 7 training bouts over 2 weeks. So you're doing essentially a workout every other day. They did muscle biopsies at rest, 3 hours after recovery, and then over the next few sessions. Notice the first time you did that workout, you get an mRNA response. Cool, you get it, right? The mRNA makes a protein. So notice you had the mRNA response, and then over the recovery period, you made a little bit of that PGC1 alpha protein. You did some more training sessions, you have some more RNA responses. The RNA goes away, but the protein keeps getting made. So over time, the more training sessions you do, yeah, the RNA response might go down, but look at the protein you've accumulated. They also looked at citrate synthase activity. Where is citrate synthase located? The Krebs cycle. It's an aerobic enzyme. If you have more citrate synthase, you're going to be better at aerobic metabolism. And look at this. The more times you do this aerobic training workout, the more PGC1 alpha you made, the more citrate synthase activity increased. Is this training adaptation? Yeah, if you had more citrate synthase at the end of the training period versus the beginning, you're going to be better at producing ATP aerobically. That's the point of it. Why? Because you had more PGC1 alpha protein, which makes more mitochondria, which makes more citrate synthase. More work with it. Yes?
Over time, the mRNA response goes down.
Right.
Is that just because you need to make the program more intense?
Well, one thing we'll learn is that the more times you're exposed to that stimulus, the less the signaling proteins get turned on. So the signaling proteins are on less, the gene expression is on. The mRNA is produced is less too. It depends on. It varies by studies, but overall, the gene expression changes depending on the training status and what type of exercise you're doing. But the gene expression response, the acute response, can go down, but also you can produce more of that mRNA at rest too. And so maybe there's less of an exercise response, but you have more at rest, so it doesn't need that much of an exercise response, right? So there are studies that have talked about this quite a bit, like why you can have a bunch of mRNA made, but only a little bit of protein made. Well, that's because maybe the ribosomes weren't put together, right? So even though you had all the mRNA, you didn't make the protein because the ribosomes weren't highly activated, or you had less ribosomes, or they've seen huge increases in protein expression, like proteins being made, with only a marginal increase in mRNA. Why? Those mRNAs were interacting with more ribosomes, which made more protein, right? So we'll talk about that in Unit 2, that relationship, but overall, you do kind of see that this mRNA response goes down with more training because you're getting more adaptive. So, overall, that's protein synthesis. Here's just kind of a review of it. These cell signaling pathways turn on from exercise. They turn on a transcription factor. That transcription factor binds to the DNA and produces an mRNA. That mRNA leaves the nucleus, binds to a ribosome. That ribosome is then translation into a protein. Okay? Resistance exercise and endurance exercise promote different signaling pathways, which means that that's why there's different adaptations to those training modalities, because they're turning on different pathways. Then there's just a little image there. All right, so these next few slides, those 4 slides here, they talk about the concepts about how do we know these proteins do this and regulate that adaptation, because we can turn one on or inhibit one and it causes an effect in the cell for some reason, right? So these next couple slides, particularly slides 12 through 16, are going to be on your exam. You need to make sure you know that, right? So we look here, muscle contraction releases calcium, right? Calcium turns on transcription factors that also interact with PGC1 alpha, makes the muscles more oxidative. We also see that when muscles contract, that tension turns on P38. P38 also interacts with the same transcription factor as calcium, right, which makes the muscles more oxidative. This example is just to show that, like, these two stimuli, calcium release and muscle tension, they're turning on the same transcription factor to produce an aerobic adaptation. Because ultimately, if you're doing lots of contractions, there's a very sustained release of calcium. And if you're doing lots of contractions, that's sensed by P38, because it's more tension, more volume, right? So both those signals, calcium signals and the tension and volume, are what's telling the fiber to become more oxidative, okay? It really ensures that our muscles are going to adapt to that stimulus in some way, rather than like, you had to have this specific response to get this specific adaptation. No, the muscle's going to try to get there and adapt the best way it can, because it listens to a lot of different signals to get there. Now, on the exam, I will never ask you what is MKK6E. I wouldn't do that. But when you look at these slides and figures, what does MKK6E represent? It's when you overactivated this protein in the P38 pathway. So wild type is just genetically normal mice. The MKK6 mice are mutant mice. It's where there was an enzyme inside the P38 pathway, they genetically overactivated. Right, so if this enzyme is on all the time, so is the P38 pathway. So what's different between these two mice? One is normal physiology, the other one is when the P38 pathway is overactive. And what do we see? When you overactivate the P38 pathway, you produce more PGC1 alpha protein. You also produce more COX4 protein expression. Would this make the muscle more oxidative? Would this make the muscle more oxidative?
Yeah.
Yeah. Okay. So these are respiratory proteins. They're involved in mitochondrial respiration, COX4 specifically. Makes the muscles better at oxidative metabolism. When mice do aerobic exercise, there's an increase in PGC1 alpha content in their muscle, and that tells the muscle fibers to become more oxidative. Now, this is myosin heavy chain 2A versus 2B, right? So, of the two muscle fiber types, 2A or 2X, which one's more oxidative?
2A.
2A, right? So, when mice do, and humans, when they do aerobic exercise, the muscles become and shift more of an oxidative phenotype, right? Why? Because these oxidative proteins build up to cause that adaptation. Now we see in response to aerobic exercise, 6 hours after exercise, so 6 and 12 hours post-exercise, there's an increase in PGC1 alpha expression in the muscle. And these mice that did aerobic cycling or running on a treadmill. We also see, though, that the expression of COX4 and ATP synthase mRNA also increased in response to that aerobic exercise stimulus. So, if we're increasing PGC1 alpha content, that's going to make more mitochondria. We also increase the mRNA, or the gene expression of COX4, which is located where?
goes.
COX-4
Oh, sorry, ETC.
in the electron transport chain, right. Where is ATP synthase located?
Same thing.
In the electron transport chain. If you have more ATP synthase, are you better at aerobic metabolism?
Yeah.
Yeah. So we can see when you perform aerobic metabolism or aerobic exercise, you have an increase in the mRNA to produce more aerobic or mitochondrial proteins. These are called respiratory genes. Why? Respiration. When you use oxygen, is that respiration? Yes. Your mitochondria also go through respiration. Why? Because they use oxygen to produce energy, right? So respiratory genes are ATP synthase and COX-4. They're in the mitochondria, and those proteins in the mitochondria use oxygen to make energy, right? So if you have more of those proteins available, you're better at aerobic metabolism. Now, what happens when you block the P38 pathway, right? I'm not going to ask you on the exam, what is SB203580? I don't do that. I don't think that's fair. But I will say, what does this slide show? It shows that when you give mice this compound, it blocks the P38 pathway. So whereas the other slide was showing you when you overactivate it, this is what happens when you block or prevent that response. So normally, saline is just salt water. It's like placebo, it's controlled, right? So they had these mice, and one condition they gave them salt water, which is no intervention, no effect. The other, they injected this inhibitor. Normally, exercise activates P38. We see that. P38 turned on was more phosphorylated when you contracted. But if you were giving them and injected the inhibitor, P38 did not turn on. Would we expect that? Yes, that's what the inhibitor does. It blocks that response, right? So it makes sense. Normal, P38 inhibited. What happens with adaptations? When you block the P38 activity from exercise, you did not increase the amount of PGC2 alpha in the muscle. Notice, the control, PGC2 alpha increased. When you blocked P38, PGC2 alpha did not increase. And also, when you block P38, you did not make ATP synthase RNA. You also did not make COX-4 RNA. But when P38 was available, you did increase that. Right? So those other slides we were saying when you overactivated that, you made more aerobic adaptations. These slides are saying when you block that pathway or that protein activity, you don't get those aerobic adaptations. So how do we know? How do we know that P38 regulates oxidative capacity? Because when you overactivate that protein, you get more oxidative proteins and adaptations. When you inhibit that protein, you get less oxidative proteins and adaptations. This is how we know it regulates that function or that adaptation. Okay? Now you might ask, is this true in humans? Why do we care about this? This is a really neat study. It's actually one of the first studies that they did, like kind of muscle, like exercise biochemistry, like muscle biopsies and cell signaling. It's like 2006, I think, actually, 20 years ago. Anyway, they took elite world-class powerlifters. This was the strength training group. Then they also took elite world-class cyclists. This is the endurance training group. But they had each of them do 2 different workouts. One was a cycling exercise bout, the other was a resistance exercise bout. So what happened? When the powerlifters or strength athletes did cycling exercise, AMPK turned on. When the cyclists did cycling exercise, AMPK did not turn on. Now, are both these groups highly trained?
Yeah.
Yes. But what are they not trained in? The same thing, right? So their powerlifters are highly trained. They are not trained in cycling. In fact, they're novice. They're not totally untrained, but it's very novel stimulus for them. They're a novice at that stimulus. So this is going to cause a large stress response in their muscle. It's going to turn on a signaling response. Why? Because they're not trained in that modality. They have no adaptations to handle this exercise stimulus. They're built for resistance exercise. But guess what? The cyclists, they have 15, 20 years worth of those training adaptations. So when they do cycling exercise, AMPK does not turn on because they've already adapted to that stimulus. This is not stressful for them anymore. What about resistance exercise? Well, when the powerlifters did resistance exercise, AMPK did not turn on. But when the cyclists did resistance exercise, it did turn on. Why? They're not adapted to lifting heavy loads. They're usually doing light loads for a long time, not heavy loads for a short time. So that's a new stress, tension-wise, metabolically, the substrates that are used, the overall intensity of the session, all those stimuli turn on these pathways to make them more resistance exercise-based. Yes?
Can that, and if you do the resistance exercise with the AMPK, there's still some mitochondrial biogenesis?
Probably not. The AMPK being turned on here is just because it's a new stressor. When AMPK turns on from resistance exercise, it doesn't really turn on mitochondrial protein synthesis the same way. It's really more just in response to maybe mobilizing substrates, or it does turn on PGC and alpha, but ultimately it doesn't turn on your muscles to make more mitochondria to the same extent as aerobic exercise. Does resistance exercise increase the number of mitochondria in the muscle? Slightly, sometimes.
So since they're...
But all the time, no. To get that adaptation, you've got to do a specific type of workout, which is aerobic training, to cause the most adaptation in that. Yes?
Since they're very like highly trained endurance-wise, would that mean that could be like still a little beneficial for them since it'd be something different?
It could be. It might help them be mobilizing more glucose, maybe anaerobic type of adaptations. You know, AMPK does make more mitochondria. It does promote anaerobic. It does promote aerobic adaptations. But there are proteins that are anaerobic that are turned on by AMPK as well, right? But they're like more type of like anaerobic type of enzymes and stuff like that. It's not. Largely the role of it is to promote aerobic adaptations. But if you're doing contractions that are very new and novel, like they're really heavy, or it's a new rep and set scheme, using a new type of substrate you normally wouldn't use right away, like glycogen versus fats, then that'll turn on AMPK in that way. You're not going to make a lot of mitochondria. Also, these are already very well trained cyclists. They already have a lot of mitochondria, right? So even if AMPK turns on, they're already at the genetic limit of their mitochondrial content. What AMPK is turning on there is maybe mobilizing glycogen, mobilizing glucose, GLUT4s, those type of things, not necessarily adaptations to make it more aerobic. Right? Same thing, P38. These cell signaling proteins turn on to the exercise mode that is novel, that is new, right? Remember, if we go back to the start of this lecture, signaling proteins turn on because they respond to exercise stressors, stress. If that protocol is new and unaccustomed, that is stressful, it's new. It's like, oh, we're doing this. What if I said, oh, actually, this is not XPhys, this is biochemistry 2. I didn't enroll in that. That's stress, right? It's new, it's unaccustomed. It's gonna be stressful. So realistically, these things turn on because when you're exposed to that stressor, this time it's gonna try to regulate adaptation. If you never do that stimulus again, well, the muscle is not going to shift to that, right? Because it was only one time. But the more times you do that stimulus, the more signaling, the more transcription, more protein synthesis, those adaptations accumulate. And now after 12 weeks, you've got a ton of mitochondria, or you've got really big muscle fibers, right? Because it's based on you doing that stimulus multiple times to get to that adaptation. And now that stimulus is now easy. And what do you do if you want to adapt? Now you got to change the stimulus again, make it harder, make the weights heavier, make them run faster or run longer, right? But this is the concept around adaptation because we're trying to keep signaling these, getting these signaling molecules. Anyway, that's up to that. So you're only responsible on the exam up to slide 16. But I will say, this slide 5, and slides 12 through 16 are going to be really important for your exam. You have to know those slides for your exam, for sure, okay? Multiple times are asked in many different ways, so make sure you know those. Anyhow, good luck on the quiz today, and I guess I will see you guys next week. Yeah, that's it. If you have any questions related to the